He will start from collagen again, okay? Because the online session was a disaster. Now, collagen here, what are we studying? Carbohydrates or fats or proteins? Proteins, okay? It's a polymer or it's a monomer or what? Polymer, okay? Made up of amino acids. Now, again, when you come to study the collagen, you will remember this picture. This is the most important picture in collagen, this basic picture. Now, tell me, Abdullah, how can you describe this single collagen molecule? It's made up of how many polypeptide chains? What do you see? Did you attend the online? Why? What's your reason? Okay, very good. Yes, Farouk. This collagen molecule is made up of how many polypeptide chains? 3. Okay, these 3 collagen molecules, how are they connected to each other? They are what, Mariam? Coiled, okay? They are twisted to form a triple helix, okay? So I'm going to study the collagen in 2 parts. I'm going to study a single collagen molecule, like the one you see here, and then I'm going to describe the whole structure. This single collagen molecule looks like this, a very simple structure. It's made up of 3 polypeptide chains, as you can see. Those 3 polypeptide chains are coiled together, looking like a rope. In biology, we describe them as having a triple helix structure. Triple because, Abdullah, they're made up of how many chains of polypeptides? 3. Helix because those 3 structures are coiled, okay? Now, every single polypeptide chain is a loose helix, not an alpha helix. An alpha helix, if you remember, in the secondary structure is a little bit more coiled. This single polypeptide chain is loose. It is made up of a repetitive sequence of amino acids, as you can see. Mainly which amino acids? Who can remember? Hydroxyproline and glycine. Glycine is very important. As you can see, glycine is always amino acid number what? Number 3. It's always positioned towards the inside of the chain. Why, Farouk? Excellent, because it's the smallest. Can you help me draw the structure of this glycine amino acid? It's like any amino acid with a central carbon bonded to an amine group, which is one of its functional groups. The other functional group is what? Who remembers? Carboxyl. This central carbon is bonded to a hydrogen. This is the basic structure of the amino acid, plus there is a variable what? R group. This glycine, its variable R group is made up of what, Farouk? Only 1 hydrogen. And this is the smallest amino acid. Okay, and this is a big advantage because this keeps the 3 polypeptide chains very close and helps us to make a tight triple helix. Okay? What? The drawing? It erases automatically. Okay? Hydroxyproline, glycine. Proline, hydroxyproline, glycine. Glycine, the variable R group is hydrogen. You have an amine group, the functional group, and a carboxyl group. Okay? Fine. Is this clear? Proline, hydroxyproline, glycine. Proline, hydroxyproline, glycine. Now, this triple structure, how is it maintained and how is it stabilized? Again? By hydrogen bonds within the structure itself. Okay, good. And then you have many collagen molecules, as you can see, that are parallel on top of each other. How are they bonded to each other? By covalent crosslinks. The name is here, by covalent crosslinks. So I have collagen, 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 and they are bonded by covalent crosslinks because they have to be attached. Do they line directly on top of each other, Abdullah? No, their ends are overlapping. This is a key term. Their ends are overlapping, or their ends are out of step or staggered, okay, in order to avoid having a weak spot. Look at this arrangement. If they were arranged like this, for example, where is the mouse? Let's say, where is the pen? You're very unlucky. Yes, let's say they are arranged like this. You have a collagen, and there's another one, and there's another one, and there's another one. And this empty space is considered in physics a weak spot. In other words, this part can easily break. So we don't want that. What do we do to avoid this weak spot? The collagen molecules, they overlap. Their ends overlap, as you can see. Okay, and they are bonded together by covalent crosslinks. Any questions? Now you have many collagen molecules, just like the cellulose. They make fibrils. Many fibrils make what? Many fibrils make fibers. Like this picture. I hope it's clear. Okay. Now, when you come to study it, organize your answer because there are so many essay questions over collagen. Why is it strong? This type of protein is globular or fibrous? Fibrous. Fibrous means it is classified or described as being long and thin and soluble or insoluble. Insoluble, okay? Insoluble in water, and it is found in lots of connective tissues, in your hair, in your nails, in your skin, etc., etc., etc. Why is it insoluble? Because most of the R groups are non-polar. And the proline and the hydroxyproline, their R groups repel each other. You know, when you have two magnets that are of the same pole, you can never approach them to each other, yes? North and north, south and south. When they repel each other, they stay fixed in this position. This helps in stability. Okay? Very good. Fine. So this is the sheet I sent you. It's very similar to what you have in the notes, but I keep on simplifying things so that, what's this called? It's easy to study. Now, the reasons for the strength of the collagen. Now, I added that. It's there, but I made it nice in points. Why is the collagen a structural protein and why is it very strong? For the 6 reasons. You will never get a question over 6 marks. So this is going to fulfill the question because it has a triple helix structure. This tight triple helix makes it very strong. High mechanical strength, high tensile strength. High mechanical or tensile strength means I cannot stretch. Look, it's flexible. I cannot stretch and it does not break. Okay, this is what it means by high mechanical and high tensile strength. The many hydrogen bonds. Now we know hydrogen bonds is found in all biological molecules. A single hydrogen bond is weak or strong? Weak. Many makes it very strong. We have lots of hydrogen bonds in water, in collagen. This increases the strength. Okay. The staggered ends prevents weak spots. I think this is clear. The repulsion of the R groups. This gives it stability. They don't ever attach. It's insoluble in water because it has many non-polar Farouk sit property. It has many non-polar R groups that do not dissolve in water, do not make hydrogen bonds with water. Okay? And they have the covalent crosslinks. Any covalent bond is actually very strong. Also, it gives it strength and stability. Any questions? Okay, now tables are very important because they help you compare and they love comparison. Now they're asking you between collagen and hemoglobin. A lot of students get confused and I highly advise you that when you come to answer a question, draw the structure. So you remember, hemoglobin reminds me, Mel, how many polypeptide chains? What does it do? What is the function? Transports oxygen. Globular protein, 4 polypeptide chains. Yes. Soluble or not soluble? Soluble. When I come to study hemoglobin, before I start studying, we'll do something like this. Okay, 1, 2, 3, 4. Hopefully, this will remind me that it has 4 polypeptide chains bonded together by disulfide bonds. It has a central heme group, like Mariam said, that carries what? Oxygen. Okay, it is soluble because the hydrophilic R groups are facing outwards towards water, creating hydrogen bonds with water. and the hydrophobic R groups are inside the center to give it what? Stability. Okay, collagen, Farouk. How many polypeptide chains? 3. Okay, I'm going to draw the collagen like this when I come to study. Not on top of each other, and do this. To remember, okay, this is not a coil, or it is a coil but made up of 3 polypeptide chains. It's also a quaternary protein because it's made up of more than 1 polypeptide chain working together as one unit. And this will hopefully remind you of everything comes next. Okay, this table is very nice. Number of polypeptide chains, 3. Here 4. 2 alpha globin, 2 beta globin. The shape, long and thin, spherical and round. Type of protein, fibrous, globule. The function, structural. Here, functional, transports oxygen. Amino acid variation, which one is repetitive, which one is variable? Collagen is repetitive, okay? Prosthetic group, is it present? Only in the hemoglobin, which is soluble hemoglobin. Okay, and I think this is done. We'll study this in circulation. This picture you have, it's a very nice and simple picture. I think we're done with this. Okay. For your information, we finished the biological molecules. We still have water. So water is very easy. Now, this is also a lovely table to remind you of the different bonds that happens in the carbohydrates and the proteins and the fats and the blah, blah, blah. Fine? Look, this lesson, a lot of students find it very difficult, but it's actually very easy when you come to organize it and study it well. You will study carbs, you will study proteins, you will study lipids, and you will study water in this lesson plus the food tests. Carbohydrates, tell yourself that I will study monosaccharides, disaccharides, and polysaccharides. Carbohydrates is the longest part. The monosaccharide, you need to know the structure of the alpha glucose and the beta glucose, okay, and the fructose. You need to know these structures. The disaccharide, you need to know how to make the maltose. Who reminds me? Maltose is what and what? What joins with what? Alpha glucose plus alpha glucose. And also you will study the structure of the sucrose, which is what bonded to alpha glucose plus fructose, okay? Lactose, you don't need to know its structure. The polysaccharides that you will study are 3. Starch, cellulose, and what? Glycogen. The carbohydrate is finished, okay? The starch, alpha glucose, amylose, amylopectin, cellulose, beta glucose, every other beta glucose is rotated, remember? You don't study, very good. Glycogen is exactly like amylopectin, but it's branched. So you summarize. Proteins, you will need to know. Collagen. Okay, the amino acid structure and hemoglobin. Collagen and hemoglobin. I think now it's easy because they're very different. Hemoglobin circular, collagen long and thin and coiled, and it's so different. Fine. Lipids, phospholipids, and triglycerides, the glycerol and the 3 fatty acids. Okay? Water, we'll talk about it now. Now, what is the bond in carbohydrates called? I'll give you 2 options because I know that you did not study. Glycosidic bond or peptide bond? Glycosidic. And the bond in proteins, glycosidic or peptide? Peptide. And in lipids is called peptide or ester. Ester. Okay? So this nice table here identifies the bonds. Carbohydrates and the lipids, they're very close. You have a central oxygen bonded to a carbon and a carbon. And the lipid the same. A central oxygen bonded to a carbon and a carbon. But how do I know that it's a lipid? Because one of the carbons is double bonded to an oxygen. This is part of the carboxyl group of the fatty acid. Okay, a quick reminder. Again, for example, maltose is of alpha glucose plus alpha glucose. The alpha glucose, in case you forgot, 6 sides, 1, 2, 3, 4, 5, 6. the oxygen is up there, and the carbon will move in a clockwise direction. Carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6 outside the ring, okay? This is alpha, so carbon number 1, hydrogen above or below? Above, hydrogen above. And then we will post the hydroxyl groups, very easy. Down, up, down. Number 1 also is down. And the same alpha glucose here, another alpha glucose. Done. This is carbon number 4. The OH is below. Here we have our the release of what? Water. So look at the bond that happens. Of course, I have here part of my screen is destroyed, but the bond here, you have a central oxygen bonded to carbon number 4 and carbon number what? 1. 1 to 4 glycosidic bond. If you notice here, the central oxygen is bonded to 2 carbon atoms and none of them is double bonded to another oxygen. But the lipids are different. Who will remind me of the structure of the fatty acid? We are revising here. The fatty acid is a chain of what, Heidi? Hydrocarbons. So I have a chain of hydrocarbons, carbon bonded to hydrogen. What are the 2 functional groups in fatty acids? Carboxyl group, yes. And the last is the methyl, CH3, okay, CH3. And it is bonded to a what? Glycerol, Zakru, please, you will forget. The glycerol is made up of 3 carbon, and every carbon is bonded to, let's use this, hydroxyl group, okay? Hydrogen, سهلة جدا. Okay, so these will join to give me what? Water. So the bond here will be a central oxygen, also bonded to 2 carbon, but one of the carbon is double bonded to what? To oxygen. So when you look and find that the carbon is double bonded to the oxygen, know that this is an ester bond. Because you will be given many bonds in paper 2, and it will be very confusing. So you have to know how to identify them quickly, okay? Now the peptide bond is very easy because amino acids are the only biological molecules that have what? Nitrogen. The moment you see a nitrogen bonded to the hydrogen of the methyl, the hydrogen of the amine, and the carbon of the carboxyl group, then this is peptide. The moment you see nitrogen, it's peptide with no negotiation. The confusion is between carbohydrates and lipids. Once you see one of the carbon atoms double bonded to an oxygen, it's an ester bond in lipids. If you don't see a double bond, then it is the glycosidic bond in what? In carbs, okay? We will study the phosphodiester bond when we study nucleic acids, okay? Water. We started water in the online. It's very easy. Tell me, what did you understand from the online? Don't look at me like this. Don't make me feel that I'm wasting my time. Water is made up of what? It's polar, dipolar. What is the shape of the water molecule? Dipolar, polar. Okay, what does polar, dipolar, what does this mean? It's made up of what? 1 what and 2 what? 1 oxygen bonded to 2 hydrogen. Polar means we have electrons being shared. Are these electrons equally shared? Unequal distribution of electrons. Overall, it has a neutral charge, but there is unequal distribution of electrons. The electrons are more strongly attracted to the oxygen or the hydrogen? Oxygen. So the oxygen will have a partially negative charge. It's called electronegative or partially negative or slightly negative. And you need to know the sign. And the hydrogen will have a partially positive. So it's going to be called electropositive. This setup makes it a polar molecule, unequal distribution of charge. Also, it is described as being dipolar because I have 1 electronegative and 2 electropositive. Okay? Now this gives it all its physical features. This bond, before I forget, is the hydrogen bond or a covalent bond? Covalent bond. This is the covalent bond within the inside of the water molecule, of the single water molecule. This water molecule is bonded to another water molecule by what? By a hydrogen bond. Thank you. Okay, now this hydrogen bond in water, you can easily be asked to draw it. Very easy to draw. This electropositive hydrogen will be bonded to what? To the oxygen or the hydrogen? To the exactly. So I will do this. This is electronegative. This is the hydrogen and the hydrogen. And the hydrogen bond is always drawn like this in dashes or dots. Clear? Okay. Now this gives it the polar nature. It makes it a good solvent. Why does it make it a good solvent? Good solvent means that any other polar molecule or any other ion can dissolve in the water. Farouk, can you tell me how does this happen? Let's say sodium chloride. Sodium chloride is an ion. When the sodium and the chloride, I think this is clear now. When the sodium and the chloride dissolves in the water, they dissociate to give you what? What do you see? Sodium and chlorine. What is the charge of the sodium? Positive. So it will be surrounded by the electronegative oxygen. What is the charge of the chlorine? So it will be surrounded by the electropositive hydrogen. When the water molecules surround the ions, then we say it has dissolved. Now it is a solution. Now the ions are free to react. Now the ions can be transported. That's why water is a transport medium. It transports everything, okay? And this is the same case for ions or polar molecules. As long as the molecule has a charge, it can dissolve in water. Clear? Because either the negative oxygen will be attracted to the positive charge or the positive hydrogen will be attracted to the negative charge. Anything hard about that? Good. Okay? Okay, it's a transport medium in all these systems of the body, in the blood, lymphatic system, excretory system, digestive system, etc., etc., etc. Now, this is important. Advantages of water. Water has a high specific heat capacity. Did you take this in physics? Yes?
Not yet.
Not yet. What is high specific heat capacity? This is the amount of heat needed to increase the temperature of 1 kilogram of water by 1 degree Celsius. Water has a very high specific heat capacity. What does this mean and why is that? Water has so many hydrogen bonds. All these hydrogen bonds have to break first in order for the temperature of the water to increase. Water is in your body. You are 70, 80% water. Why are you always 37? For so many reasons. And one of those reasons is that the water in your blood has a very high specific heat capacity. It needs lots of heat for your temperature to rise from 37 to 38 because it needs lots of heat to break the hydrogen bonds. This makes water very stable. It stays in the liquid state for as long as possible. And this is a big advantage. It's the biggest advantage because the water stays liquid in your body. If the water changes quickly to solid or to gas, what will happen to you? Karsa, okay? And the same for plants. Water stays in the liquid state inside the plant's body for the longest time possible, okay? And the same for fish, surrounded by water. Throughout the year, it stays in this liquid state, okay? The sea does not freeze up. The sea does not all evaporate. Because can you imagine the number of hydrogen bonds in the ocean? All these need to break for the whole ocean to evaporate, which is nearly impossible. Clear? Another advantage. It has a high latent heat of vaporization. Again, physics. What is latent heat of vaporization? To change from what? Exactly. Okay. How can this be explained? Very simply speaking, this is your skin and this is a drop of sweat, which is mainly water. This sweat will evaporate eventually. It will change from the liquid state to a gas state. In order to change from liquid to gas, it will need to absorb lots of heat from your body to do that because this heat will be needed to break the hydrogen bonds in the water. So as it evaporates, it will absorb a lot of heat from your body, cooling you down. So it is a good cooling agent. It helps to cool you down. It cools down your body. It cools down the body of plants, of animals. As the plant transpires, it takes heat and it evaporates. So it cools down the plant. And you cool down when the sweat evaporates. Clear? This is latent heat of vaporization. Fine, it reduces the chance that water will freeze. Yes, also. If you have water in the liquid state, in order for the liquid water to change to solid, it also needs to release lots of heat to the environment so that the bonds solidify. And this is not easy. So again, water stays in the liquid state for a very long period of time. It's not easy for the water to evaporate, and it's not easy for the water to become solid. To evaporate, it needs to absorb lots of heat from the surroundings to break the hydrogen bonds. And to change from liquid to solid, it needs to release so much heat to the surrounding. Okay, again, in order to combine the hydrogen bonds and make them change from the liquid state to the solid state. This means that the water will not freeze easily. And this is a big advantage, again, because you have water in your body. This water stays liquid. It doesn't evaporate easily, and it doesn't solidify easily. This water is in the sea. Imagine the water all solidifies in the sea. All the fish will die. Clear? Okay, good. Now, the unusual density and freezing properties of water. Again, this is a little bit of physics. You know that ice has less density than water? Ice floats on water. Do you know the icebergs? Okay, so you have a sea and you have icebergs. They're floating on water. Why? Because they're less dense. And this is also an advantage because it will insulate the water underneath and keep the fish warmer. Also, when the water at the base of the sea solidifies, it will move up because it's less dense. This movement of the ice creates currents in the sea, and this is also good for the aquatic animals. Solid ice is less dense. Below 4 degrees, the density of the water starts to decrease. It floats and insulates the water below it. Advantages: large bodies of water do not freeze, increases the chances of life surviving in cold conditions. Okay. Changes in water density with temperature causes currents, and this helps circulation of nutrients in the ocean. In other words, water is a liquid. It's very difficult for water to be gas or to be solid because of the hydrogen bond that either need to break or either need to make. Clear? Tamam? Good. Cohesion. It's the attraction of similar molecules. Water attracts each other. And this is an advantage. Okay, again, how do they attract each other? By hydrogen bonds. This is an advantage because it helps the water to move up the xylem. Okay, every water molecule will pull the water molecule below it. Okay, also, it helps animals to move on the surface of the water because the surface of the water attracts each other. It makes like a layer. Okay, when you put a drop of water on the desk, doesn't this drop of water curl up? Okay, because the water molecules are attracting each other. This is cohesion. Fine, it's a big advantage in plants. It helps the movement of the water up the xylem. Fine? No. Water also attaches to other molecules. This is called adhesion. Again, we'll study this in plants because the water adheres to the wall of the xylem so that it moves up the xylem. And the water, every water molecule pulls the water molecule below it. These two forces help in the movement of the water up the xylem. Okay? Okay, Hedy? Water is very, very easy. Okay. Again, advantages of water. It's a solvent. And this allows chemical reactions to occur. It's a transport medium due to the polarity of the water. The reason. It has a high specific heat capacity. In other words, I need lots of heat to change its temperature by 1 degree, which is a big advantage because the temperature doesn't fluctuate inside your body. Again, due to the many hydrogen bonds in water, presence of many hydrogen bonds, okay, allows a water to be suitable habitat. Again, where is it? Go back. High latent heat of vaporization. I need lots of heat for the water to evaporate. So when it does, it will cool you down in the process. It's due to the presence of many hydrogen bonds, it's a good coolant. Anything hard about water? I'm done. I'm finally done. Okay, what will we do till the end of the session? Get out paper 2. We'll solve together. Oh. Hedi, if you don't have your classified or you have it? Okay. Okay, I don't have a voice today, so you will read the question and you will tell me the answer. What's this called? Open your class. What page is that? 73. Quickly. Tell me, Abdullah, what's A? The molecule formed from a condensation reaction between fructose and glucose. Sucrose. Okay. Condensation involves what? Release of what? Of water. Okay. So sucrose is not wrong. Sucrose. Okay. The name of the bond broken when two amino acids are separated by hydrolysis. What's the bond in proteins? Peptide. The unbranched polymer consisting only of beta glucose molecules. Cellulose. Excellent. The reagent used to test for the presence of proteins. Burette. The molecule produced in addition to fatty acids when a triglyceride is hydrolyzed. The molecule produced in addition to fatty acids when a triglyceride is hydrolyzed. You're breaking a triglyceride, fatty acid and glycerol. Yes. Okay. Okay. On the exam, you will read the whole thing. And I'm not going to. Describe two ways in which the structure of a collagen molecule differs from the structure of an elastine molecule described above. No, you have to read. So read the elastic fibers and let us read. Elastic fibers are made up of bundles of fibrous protein. Each molecule is a single. Okay, if I am you, I'll make a mark because collagen, how many polypeptides? 3. Okay, so single here. The primary structure contains a large proportion of the amino acid glycine. This is a similarity. The glycine has a hydrophobic R group. Okay, this is a similarity. They're talking about glycine. Glycine does not occur at regular intervals. This is a what? Does not occur. In the collagen, it does occur at regular intervals. Every third amino acid is glycine. The polypeptide forms random coils that change shape as the elastic fibers are stretched and recoiled. The collagen, are they random coils? No, they are a stable triple helix. Elastine molecules are joined to each other by many covalent bonds to form cross-linked. This is a similarity. So tell me, Karim, what are two differences in what you read? And then Karim, this is going to continue this attitude of yours? Farouk, tell me what are two differences between collagen and elastin? Very easy question. In the what? Identify 3 polypeptides where? In the elastin or the collagen? In the collagen. What else difference? Regular intervals. Okay, every third. Can you give me a third difference? What you said is correct. Give me a third difference. It's not random coils. Collagen are a fixed triple helix structure. Okay. You want to look at the mark scheme? Yeah. ايه يعني accept؟ ايوه accept sucrose. ايه ما انا ما قلتش، ما انا كنت بفكر يعني معاك. هو في ووتر بيطلع بس هم عايزين السكروز. What? No, purine. Okay. You can write its composition or just purine. Fine. here, collagen has 3 polypeptides. We said that. Glycine is every third amino acid. We said that. So we will take the 2 marks. Okay? Good. Fine. Draw the ring form of alpha glucose in the space provided. Then this means that you have to memorize the alpha glucose or not. When you are asked to draw a single ring, then you draw all the elements, the hydroxyl and the hydrogen and blah, blah, blah. Take a minute and try to draw it. Don't look at the mark scheme. يلا تفضل. And then I'll draw it. Kareem, where is your classified? Huh?
And the first carbon of the
Above. Oh. Okay. How many sides do I draw? Help me. 6, 1, 2, 3, 4, 5, 6. Oxygen is up there. This is carbon 1, 2, 3, 4, 5. CH2, OH. I advise you to go to carbon 2 and start from there. I'll do the hydroxyl down, up, down. Okay? And then go to carbon 1. This is alpha or beta? Alpha. So the hydrogen is above and the hydroxyl is below. You can do the hydroxyl groups only, but I advise you to just put all the elements to be on the safe side. Let me use a different color. Okay? Now I'll add the hydrogen. The first carbon is done. Second carbon, the hydrogen is in the opposing direction. That's okay. So either put all the elements or just include the hydroxyl groups, but at least the first hydrogen in carbon number 1 is important. Fine. Name the type of bond formed for a covalent bond between 2 alpha glucose. What's the bond between 2 alpha glucose molecules? Between 2 alpha glucose molecules. صح alpha شابترين ايه ده؟ alpha 1 to 4 glycosidic bond. Okay. Okay, we still did not take protein synthesis, but what is a gene? You know it in the O level. Length of DNA that codes for a particular protein or characteristic. Write it, write it. Let's finish. Length of DNA that codes for a particular protein. Okay, this table is cell structure. Which organelle assembles ribosomes for polypeptide synthesis? In other words, which organelle makes ribosomes? Nucleolus. Ribosomes are made in the nucleolus. It assembles ribosomes for protein synthesis. Okay, it's made in the nucleolus. It synthesizes ATP to produce. I will not continue. What synthesizes ATP? Mitochondria, of course, very easy. Folds and modifies polypeptide. What does the modification of protein? What did you say? إيه؟ حرام عليك يا نسي. The lysosome, what's lysosome? The lysosome is an organelle that contains hydrolytic enzymes that kills or breaks down anything not wanted by the cell. We will modify the protein in the rough endoplasmic reticulum or Golgi body. These two modifies proteins. Remember, we make the protein in the ribosome, and then it goes to the Golgi body, and we add a protein, we add a carbohydrate, we add a lipid, whatever. Okay, now, you see this, what's it called? They tell you it's an enzyme. So this is which type of protein? Primary, secondary, tertiary. Enzymes are what? Tertiary, okay? And this is which bond? Disulfide, very easy, okay. Name the 2 types of covalent bonds in the structure. Bro, peptide between the amino acids and the disulfide. Good. Peptide and disulfide. Next. Explain the meaning of the primary structure. 1 mark. That's it. The sequence, the order of amino acids. Okay. Who determines this order of amino acids? The DNA. Okay. What is meant by hydrolysis? Yes, to break a what? A hydrogen bond, a covalent bond. The term hydrolysis, addition of water, to add water, okay, to break a covalent bond. Suggest the type of biological molecule which is the substrate for lysosome, lysozyme. Lysozyme is an enzyme. What does it break? 1 minute. Definitely, look here. The lysozyme. Type, I have it here. I have battery. I don't have battery. Alhamdulillah. The lysozyme hydrolysis compounds found in bacterial cell walls. What is found in the bacterial cell wall? The bacterial cell wall is made up of what? Peptidoglycan. ذكروا.